You know, deep inside every single one of your cells, there's a kind of genetic control panel that's making decisions moment by moment. So today, we're going to pull back the curtain and find out how our bodies decide which genes to switch on and which ones to leave off.
Okay, so here's a mind bender for you. Every cell in your body, from a neuron in your brain to a cell in your big toe, has the exact same DNA, the exact same instruction manual. So how on earth do they end up with such completely different jobs?
Well, it turns out the secret isn't what's written in the genetic code, but which pages are actually being read. It's a process we call gene expression. Imagine your DNA as a giant cookbook. A muscle cell just opens it to the recipes for building proteins, while a brain cell flips right to the chapter on sending electrical signals. The rest of the book, it stays closed.
And here's where it gets really wild. The world around us can actually flip these genetic switches. A study on macaque monkeys found that their social rank, their place in the pecking order, directly changed which of their genes were turned on. The high-ranking females literally had stronger immune systems because their status was activating protective genes. It's incredible proof that our DNA isn't some static blueprint, it's a dynamic script that our lives are constantly editing.
Okay, so how does a cell actually manage this unbelievably complex dashboard of switches? For organisms like us, it's not a simple on-off button, oh no. It's more like a high-security system with multiple layers of control. So let's break down the 5 main levels.
You gotta think of this as a whole series of checks and balances. First up is just physical access. How tightly is the DNA packed? Can we even get to it? Step 2 is the master switch. Do we even start reading the gene? Then, step 3 is like an editor, cleaning up the message. Step 4 is the volume knob, deciding how much protein to make. And finally, step 5 is the last quality check, activating the finished protein so it can go do its job.
So let's start with step 1, the packing. Think of your DNA like a giant scroll. When it's loosely unfurled on a table, that's what we call euchromatin. It's active, it's accessible, it's easy to read. But when that same scroll is wound up super tight and locked away in a chest, that's heterochromatin. The information's still there, but it's hidden and turned off. You just can't get to it.
And you've probably seen a perfect real-world example of this, a calico cat. See, female mammals have 2 X chromosomes, but they only need 1 working in each cell. So, early in development, each cell randomly picks 1 X chromosome and just packs it away super tightly into that unreadable heterochromatin. And because the genes for black and orange fur are on the X chromosome, this random shutdown creates those beautiful, unique patches of color. A calico cat is literally a walking, purring map of gene silencing. Now, to really get a handle on our own complicated system, it's super helpful to look at something a bit simpler first. Let's turn to bacteria. These guys are the absolute masters of efficiency, and their genetic switches, which are called operons, are the perfect way to learn the basic principles. So the whole genius of an operon is that it's just so streamlined. Instead of having a separate switch for every single gene needed to do one job, like digest a sugar, bacteria just group all those genes together. Then they control the whole set with a single on-off switch. It's a beautifully simple, all-or-nothing system.
And these bacterial switches basically come in 2 main flavors. You've got your repressible systems, like the tripoperon. These are on by default. The cell is always making tryptophan, which is an essential amino acid, until it senses there's already plenty floating around. Then, tryptophan itself flips the switch off. Then there are inducible systems, like the lactoperon. These are off by default. The cell doesn't waste energy making lactose-digesting enzymes if there's no lactose around. The switch only flips on when lactose itself shows up.
All of this is managed with 2 simple inputs. Negative control is basically the brake. A repressor protein literally sits down on the DNA and acts as a roadblock, blocking the gene from being read. Positive control, on the other hand, is the accelerator. An activator protein comes in, binds to the DNA, and helps get the whole process started, ramping up production. Just like driving a car, you really need both a brake and an accelerator for fine-tuned control.
All right, so we've seen how this elegant control panel is supposed to work, but what happens when it breaks? Well, as you might guess, errors in this system are at the very heart of a lot of human diseases.
We call these errors gene mutations. And these aren't just temporary glitches, they are permanent changes to the DNA sequence. Sometimes they're just random typos that happen when a cell divides. Other times, they're caused by things in the environment like radiation or certain chemicals. And the effect of a tiny change can be just staggering. I love this description. It's like a single typo in a 3 billion-letter book. Take sickle cell disease, for example. It's caused by just one single incorrect letter in our DNA. That one typo is enough to change the hemoglobin protein, which then warps the shape of red blood cells and causes a devastating, life-threatening disease. All from one letter.
Cancer is maybe the ultimate example of broken gene regulation. The runaway car analogy is perfect. You've got genes that are supposed to tell a cell to grow, that's the gas pedal. Well, they can mutate into what we call oncogenes, which is like the gas pedal getting stuck to the floor. At the same time, you have other genes that are supposed to stop growth, the brakes. If they become faulty, they're called tumor suppressors. So now you have a car with the gas pedal floored and the brake lines cut. The cell just divides and divides and divides, completely out of control. So far, we've talked about what flips the switches. But for this last part, let's ask an even bigger question. What if your life experiences could physically reach in and modify the control panel itself? This is the incredible, mind-blowing world of epigenetics.
To really wrap your head around epigenetics, let's go back to that simple question about identical twins. They're natural clones, right? They have the exact same DNA sequence. So why do they end up with different personalities, different habits, and even different risks for certain diseases as they get older?
The answer is epigenetics, and here's the key. These are changes that happen without altering the DNA code itself. The letters stay the same. Instead, chemical tags get added on top of the DNA, almost like little sticky notes. These notes tell the cell's machinery whether to read a certain gene or to ignore it. And what's more, these notes can be passed down when cells divide. And this is where it gets personal. Studies have shown that when they're just 3 years old, identical twins have epigenetic patterns that are nearly, well, identical. But look at the difference by age 50. After decades of different diets, different lifestyles, different experiences, they've each collected a unique set of these epigenetic tags. Their gene expression has diverged, which can explain why one twin might get a disease while the other remains perfectly healthy. One of the main ways this happens is through a process called DNA methylation. Sounds technical, but the idea is actually pretty simple. A tiny chemical tag gets attached directly to the DNA, and that tag acts like a big "do not disturb" sign, telling the cell's machinery to just skip over that gene entirely. In this way, our experiences can literally add annotations to our genetic code, silencing certain parts of it. So what's the big picture here? The main thing to remember is that our cells aren't just slaves to their DNA. They're constantly interpreting it. From the physical way the DNA is packed away, to all the layers of control, all the way to those epigenetic sticky notes left by our lives. It's a dynamic system. And while a breakdown in that system can lead to disease, the really powerful idea is that our environment is actively shaping how our genetic blueprint gets read every single day. And that leaves us with one last fascinating question. For the longest time, we kind of thought of our DNA as our destiny, right? Fixed, unchangeable. But if our experiences, our choices, and our environment can reach into our cells and change the very way our genes are expressed, well, that opens up a whole new way of looking at nature and nurture. It makes you wonder, doesn't it? Who is ultimately in control?